All right, so thanks so much for the chance to be here, and I have nothing to declare. So this area is really about seeing how much more information we can get from our data if we start to look at how signals and networks are changing over time. And just to start with a bit of overview, So as a bit of background, the goal of functional connectivity is to see how brain areas may be interacting as systems or networks. And to infer this using our functional MRI data, we turn to the time courses of these different regions, and we look essentially at similarities. And so if we see that there are some regions that have similar fluctuations in their activity, then maybe we might suppose that there's some direct or indirect interaction happening between them, that we'll come back to that assumption later on. And so by mapping out the relationships in these time courses, then we might try to learn something about how brain function arises from these distributed systems. So as we've seen, a core finding in this literature is that there are certain very stable patterns in the brain and in functional connectivity that we can identify over different scan sessions and over different individuals, and even if we don't scan people with the tasks in the resting state. So these are the resting state networks. And if we focus on this one called the default mode network, we can still identify it to a large extent as a correlated network, even during sleep, and in monkeys, and in rats, and even under anesthesia. But if we look at these things more closely, we can see that the connectivity of various brain regions may also change under different contexts and conditions. So for example, in deep sleep, the default mode network seems to show a decoupling between the anterior and posterior parts compared to in wakefulness. Or depending on what you're doing or thinking during the scan, this may introduce a different pattern of region-to-region correlations. There may also be changes in connectivity with neurological disorders or neuropsychiatric disorders, and this was a key early study by Michael Grecius looking at finding that in Alzheimer's disease, the hippocampus becomes less strongly correlated with other parts of the default mode network compared to in age-matched controls. So if we look at the variability of connectivity under these different conditions, then this may give us a useful window into how systems are changing to support these different types of functions and how they may be deviating in pathology. So this is a scan that was 54 minutes long. It's kind of an unusually long resting scan, since these are usually about 15 minutes at the most. And this is the matrix you get if you take a set of regions and you look at correlations between them over the entire scan. And we can look at what happens if we start to break this down into smaller time intervals now. So here's the first 30 minutes, the last 24 minutes, and then if we break those up even further. So we noticed that some of these matrices look quite similar to each other, but then we might have one or two that look different. And we might wonder, why is this? So is this just something related to noise and something we shouldn't care about? Or is there something valuable that we can look at in terms of this temporal variability and whether there are different states or modes of activity going on? And we can also see from this picture that if we make only one matrix of connectivity, we only know then about the average characteristics of connectivity. We don't have any access to the information about things that may be varying over time. All right, so if we look at our data, we can see these time-varying changes happening, but what can this tell us about brain function? What is a little more motivation for why we might do this? And first, it's been known that there are it's known that there's rapid changes in neural interactions. We know this from direct recordings of neural activity, and in fact, there's a longer history of studying dynamics or changes over time in electrophysiology compared to in fMRI. But when we're looking at the brain with bold fMRI, we're seeing this response that's being blurred by, in space and time by the hemodynamics. And so we don't have access to a lot of those similar phenomena. But there's been some simulation work suggesting that we might get a range of different states in our bold signal emerging from these faster dynamics. And perhaps if we look at the features of these windowed correlations, then over time scales of like seconds to tens of seconds, then we might be able to give some information beyond the average that may be useful as biomarkers. Another aspect of brain function we might study with this dynamics are changes that happen naturally more slowly over time, like emotion or vigilance and other sorts of things. And by looking at what connectivity changes accompany these, we might learn more about the underpinnings of these natural state changes in terms of large-scale brain networks. But this is all still a very new and exploratory area. So, so we can see this. The idea basically behind dynamic analysis is that instead of taking all of these time points across our scan and using that to make just one representation of the brain's connectivity, then why don't we instead try to model how both connectivity or any other features of our fMRI signals are playing out over time, and to see whether this can give us a more complete view of brain function. So now I'd like to go into some of the methods used to analyze the data in this way. And I'll start with a sliding window analysis, because this is probably one of the most direct ways to begin looking for changes over time, and it's the basis for a large number of approaches. So the basic idea here is that we can divide our scan into overlapping or not, doesn't show up, or non-overlapping, these are supposed to be covered by windows, windows of time. And then within each window, we can calculate some measure of interest, like the correlation between areas or ICA or other sorts of network measures. And then as an example of what we might see if we run this kind of analysis, this is 3 separate 1-minute windows of time. Looking at both pairwise correlations as a matrix or as a network or graph. And you see some differences. So like in this window, it seems that the network is more densely interconnected than over here. And here's what what a sliding window seed-based correlation map looks like. So each frame of this movie is a 2-minute sliding window moving over time, and we're looking at correlations seeded with the posterior cingulate cortex, which is a default mode network region. And again, you can see that there's some changes happening, regions that seem to go in and out of coherence with one another. The blue areas are negative correlations and the red are positive. But at the outset, if we start looking at these things, it's really hard to know if this is useful or what this is actually meaning, because we don't really have a good sense of what's driving these kinds of changes, and especially in resting state data. So, so it's easy to just run a sliding window analysis, but there are a number of trade-offs that we have to be aware of, and one of them is the choice of window size. So on the one hand, we'd really like to have windows that are short enough that we have sensitivity to detect change or look at transient behavior. But on the other hand, we also want to have enough time points within our window to be able to actually estimate our quantity of interest with some reliability. Otherwise, if we shorten our windows and we don't have enough measurements for what we're trying to look at, then we can get a whole bunch of spurious variability just because we have low statistical power. Another issue when we start to open this time dimension with sliding window or with any other type of analysis is we're opening up this now much larger space of features. And so how can we deal with this information? How can we summarize or parameterize it in useful ways? So to talk more about this for a minute, suppose you have this hypothetical measure of functional connectivity between regions, either calculated with sliding window analysis or from another type of model. So based on this, we can try to think about what kinds of features might be relevant to brain function. So maybe it's something relating to the overall amount of variability that regions are showing, or maybe dealing with how rapidly these changes are happening. And maybe we'd have a hypothesis like patients with depression are less variable or less flexible in terms of their changes in correlation over time between certain networks. And so just as we can do with any other kind of functional connectivity measure, we can also try to compare these dynamic metrics against conditions or brain regions and behavior and so forth. So this is an example looking at which areas in the brain or which pairs of regions tended to show the most or least stable correlations over time in their spontaneous activity. And they find that the most stable correlations tended to involve the left-right bilaterally homologous areas, and that in contrast, more variable ones were connections between non-homologous regions, particularly here, and as well as, if you think back to our resting state networks, it was the connections between those different networks that were more variable, and within the networks they were stable. And what we did here was to make a seed-based variability map, essentially. So instead of looking at the areas that are most strongly correlated with a given seed region, we can instead map the overall amount of variability displayed by a region with respect to a seed, which was the PCC here. And what we found here was that the areas with more variable connection to this PCC included salience and executive control networks, which we thought was interesting because it's been suggested that these networks may exert a modulatory influence over the default mode network. And as was just mentioned, another way we can try to find structure in fMRI dynamics is to see whether there are certain patterns of connectivity that tend to reliably recur over time. And this was an idea from Elaine Allen and Vince Calhoun. And so what you do here is you take a sliding windows of data, and you make correlation matrices between regions of interest or between functional networks. And then to see if these series of matrices have some structure, you can put them into a clustering analysis and see how they group together. And these were the types of states that they found emerging in their data. And then you can go farther and ask what the relevance of these different states might be. And so in the study looking at schizophrenia, comparing schizophrenia to healthy controls, they find that if you look at the proportion of time that the individuals are spending in one of these given states, that distribution was different between these two groups. And so again, this is another demonstration that we may be able to pull more information out from our data by looking at these dynamics. So as an extension of this method, what if we try to move from sliding windows and down to single time frames? And to introduce this idea, I think it's helpful to consider the fMRI scan as if it were a movie. And so this is someone's brain activity in the resting state. Their eyes are closed. And this is not a sliding window movie this time. This is actually just showing the normalized bold signal at each TR. And you can basically think of this as being a sequence of these frames, which are essentially multivariate snapshots of activity. And again, one way to get a handle on whether there's some structure or information in this type of data is to apply a clustering analysis. And so if we look at a scan going on here, and we start picking out some different time points, we might see that over here, here, and here, we get patterns that are, these are single bold volumes that look something like this. And then if we look at different time points, we might get other ones, and then if we cluster these, then we can see these type of representative patterns that seem to be coming up over again in our data. And this was a time course from the PCC, and I think it's interesting to note that what we do here is we're taking peaks of that signal, and so the PCC tends to be high in all of these frames that we're looking at, but what it's coactive with is different. And so over here, it looks more like the canonical default mode network pattern, but here it can look like there are other areas that are coactive with the PCC, and here more negative correlations. And so this lets us get a more time-resolved picture of what's going on in our data, and if we were to just take an average correlation map, then we would get some mixture of these. Looking at these coactivation patterns can also give us a potentially useful set of new metrics for quantifying different state changes and so forth. So here what you might do is compare resting state to a working memory task. And ask questions like, what are the most representative states or patterns in each of these conditions look like? How many different patterns are required in order to capture the complexity of the data in each of these different cases? And so what we found here was that in this more constrained, focused working memory task, there seem to be fewer and less diverse patterns compared to if we look just at the unconstrained resting state. We can also ask about sequences of patterns. So are there repeating spatiotemporal patterns that can be detected? And so here was an example of one such pattern that was found by this group. And the way that they went about finding this was to build this basically template matching approach to look for occurrences of repeating patterns. And there have been many different approaches suggested to doing dynamic connectivity analysis. These are just a few different examples. So change point analysis, for example, tries to find at what time points in the data there seem to be marked changes in the statistics of your signals, rather than just looking at over some fixed window size. And hidden Markov models and other such approaches are now being applied more and more to look at states. Changes. So now I wanted to go more deeply into what kinds of issues might be involved in terms of the analysis and how we might try to interpret some of these dynamic changes. So when we make these dynamic maps and we see that things are changing, how do we know that these things are interesting? It turns out we have to take a lot of care with this, and one reason is because as Molly already introduced, the bold signal is a very complex thing, and there are many factors contributing to what we see, and one of them is unfortunately artifacts. So we have breathing and head motion and all kinds of other things going on, and these can create non-neural variation in our connectivity measures. And so this is looking at breathing, and what's going on here is that if someone takes a deep breath, then what you see in the fMRI signals across many areas of the brain is a large and delayed bold signal dip. And this is problematic for analysis, static and dynamic connectivity, because this response is not related to the local neural activity of these regions, but rather it's being induced by changes in the arterial carbon dioxide and cerebral blood flow, which is changing this bold contrast. So unless we were recording the physiological data and knew that there was an event like a deep breath, then we might say there's this global change in network connectivity that lasts for up to tens of seconds after someone takes a deep breath. This is actually something that really worried me early on when I was starting to look at dynamic connectivity. So we had been recording cardiac and respiration data during the scans, and as I was making sliding window maps just to look at what happens, it seemed that every time someone took a deep breath, then you'd see this large increase in coherence across much of the brain. And so, spent some time thinking about physiological noise and how we correct for this, because otherwise it's hard to know that we're actually studying neural activity. Another thing to point out is that you can see variability just in random noise. So these are actually two fMRI signals that I extracted from brain regions in real data. And if you make a sliding window correlation, then you can see that they're fluctuating in some way. If you just take some white noise and convolve it with an HRF, you can also get similar amounts of variation happening. And so just because we're seeing this variation doesn't mean that it's automatically interesting. Things like this point to the fact that we really need to be testing the features of interest with the appropriate sorts of null models. White noise is probably usually not the best null model for fMRI. We really want to get to ones that match the properties of our data better, and that's been discussed in these papers. Another way we can get spurious effects is relating to the interaction between the sliding window size and the underlying frequency content of our signals. So as a little simulation, these are just two sinusoids, and they're offset from one another by just a small phase shift. You can't really see it here, but since this is a constant relationship, we'd really like our analysis to say that the relationship is constant. And if we start to use larger windows, or windows that are like magic multiples of the frequencies, then we see very small variation in the correlation coefficient over time if we were to slide this window along. But if we are using windows that are much shorter than the period of in the data, then we can get quite a bit more variability. And so papers like this have suggested that maybe something we want to do is to bandpass filter our data in accordance with the sort of window size that we want to look at. But then another question is, what type of window, what size of window do we want to choose for our data? As discussed earlier, we have this trade-off between detection and, we want to use short windows perhaps, but this would give us fewer data points per measure. Another thing we have to consider is that bold signal has this broad power spectrum. So it's sort of this 1/f-like thing, and filtered by the hemodynamics. And so we have contributions from a lot of different frequency components. And so it's not obvious at first which one we want to focus on. And when we start to choose a window, then we're tuning our, certain window sizes are better tuned to looking at certain frequencies than others. And so the time scales we might want to focus on may be very, we might have to, this may be something that depends on the particular question that we're asking. But another thing you can do is to actually just see what happens across many different time scales or frequency scales at once. So this is a time frequency analysis, and what we're looking at here is the coherence between brain regions as a function of both time and frequency. And so coherence is indicated by the colors. You can also put on some phase information with arrows if you'd like. And when we study this using a wavelet transform, one of the nice properties here is that it naturally matches the time scale of our dynamic analysis to the frequencies in our data. So the higher frequencies involve more compact basis functions, and so we're kind of looking at shorter windows versus in the lower frequencies when we need to use larger stretches of data in order to estimate how connectivity changes. But this is very nice for giving us a very comprehensive picture of what's going on, but it's also very, it's also a lot of information that we're getting, because for every 2 signals we might have this huge matrix. And so here too we might want to think about how it might make sense to reduce this to a smaller number of manageable features. So the last consideration that I wanted to discuss in this section is whether functional connectivity is, in fact, the best description of our data, or if in some cases it's equally important or more important to actually look at what's going on in the raw signals themselves. And so just a little illustration. This is, we have 2 time courses, and then we have an increase in the amplitude of these signals relative to the noise floor here. And so our windowed correlation will probably detect a bigger correlation, but we might agree that the more relevant or basic feature going on is this event rather than the correlations between the signals. And similarly, there are other things that can happen in our signals, such as changes in the autocorrelation, or these are spatiotemporal kind of traveling waves that have been pointed out. And these might all manifest as changes in correlations or other measures of functional connectivity, but may be better described in terms of other things. So in the last part, I just wanted to talk now more about how we can try to learn what kinds of factors are giving rise to these dynamics more in terms of neural activity. So if we have pure resting state data, this is hard because we haven't set up really any particular conditions or behaviors to compare. But it can be really helpful to record other things during our scan, like EEG or physiology, and then to try to see how these types of measures relate to our fMRI signal dynamics. And this may tell us something about how certain natural spontaneous state changes relate to functional connectivity. So this was a study where we recorded EEG at the same time as the fMRI scan. And so here I looked at EEG power in different windows across the scan into 2 different frequency bands. And I focused on the alpha and theta bands here because they have a known link with brain arousal state, or how alert or drowsy your subject may be. And so if we look at, if we focus on certain windows and try to make maps of the fMRI functional connectivity at those same windows, then what we found here were that at times when the EEG indicated that the subject was likely to be drowsy, we saw more increased positive correlations between our networks, and as well as more globally, compared to times when the EEG indicated that the subject was probably more alert. And so if we're looking at correlations between networks and looking at how those change over time, to some extent they may be linked with vigilance states. Dynamic correlations have also been found to track sleep stages, and so here what they did was use a support vector machine classifier to map fMRI correlation patterns and different types of EEG-defined sleep stages. And so then if you've trained this classifier, then you can take some data set and you can look at sliding window correlations as time goes on and try to use the classification to determine which sleep state the subject was likely in. And so when they did that, they got a result that matched very closely what they found with the EEG. So that's very cool because it means that we may not, in fact, need to go through the trouble of recording EEG to determine what sleep stage a person was in or if they were likely asleep. Actually, I think in this study they also found that in resting state data, after like 3 or 5 minutes or some really small number, like a lot of subjects were sleeping. So it's kind of important. There also seems to be a connection with mind wandering, probably also something that happens a lot in our scans. And so, so you might be wondering, how are they able to tell if someone's mind was wandering? And the answer was that they just asked them. So every once in a while, they would interrupt the subject and say, like, how much was your mind wandering? And so what they did was they looked at the amount of variability in the connectivity between PCC and this subnetwork of the default mode network, and found that there was a relationship there between increased variability between those networks and increased amounts of mind wandering. But they had pointed out that if you took the static average relationship, then you wouldn't see a connection here. We can also experimentally induce changes in connectivity, so bring back some kind of task. And so what they did here was look at sad movies, and they were looking at the changes in emotion in the subjects and looking at what kinds of changes in functional connectivity happen to be going on. And here they are giving an infusion of a drug. And so then we can try to learn how these brain networks are perturbed by these particular manipulations. Okay, so in summary, dynamic descriptions may provide complementary information to static functional connectivity. We may be able to study how large-scale networks are reconfiguring under natural state changes or specific manipulations, and that these things may give us biomarkers, perhaps, for health and disease. It also may help us to understand why we see so much inter- and intra-session variability in our fMRI scans just routinely. However, there's a lot of challenges. This is kind of a complicated area. For bold fMRI, we have limited temporal resolution. We have trade-offs between window sizes and SNR. And when we look at dynamic analysis, this is much more sensitive to noise than if we're doing a lot of averaging, because any little thing that happens might look like a connectivity change. We're not getting an increased robustness by doing some more averaging, and so we need really careful analysis and noise reduction and statistics to test whether these things are real. So here's just a handful of review articles that have covered different aspects of the field, and thanks a lot to people who have been very helpful in this.